Good morning, class. Just 19 of you. Okay, let me start with what we did last time. So, can one of you tell me what is the difference between the chromosome and the plasmid? Anybody? Raise your hand, students. Bhavya, bolo.
They are smaller in size and have selective advantage.
Which ones are smaller in size?
Hmm?
Which ones are smaller in size, Bhavya?
Plasmids.
Okay, go ahead.
And they are the extra chromosomal genetic material, and
Extra.
they are
Okay.
cir- they
Circular
are
तो दोनों.
mainly used in bacteria for manipulating.
Okay, Vishantan.
Man, basically they are like they provide selective resistances to bacteria, and they replicate independently of other genetic material like chromosomes.
Satyakam.
Ma'am, basically plasmids are found in prokaryotes, ma'am. Rather than saying in bacteria, we can say they are found only in prokaryotes. However, chromosomal DNA are found in both of them.
Okay, so there are exceptions wherein we find plasmids also in eukaryotes like yeast, but then, like I said, there are exceptions. So a cell, what I want to clarify here is that a bacterial cell can survive without the plasmid as well. It can perform all its normal metabolism without the presence of a plasmid. A plasmid comes into picture or is of importance when there is certain kind of stress outside or in the environment. That is where the plasmid would confer some advantage to the cell. So a cell possessing the plasmid will have a better chance of survival compared to one which does not. Okay. What else? So we covered in the last class structures which are found in the cytoplasm of a prokaryotic cell. I intend to cover all the other structures today of a prokaryotic cell and move on to the eukaryotic cell from the next class. Let me share. Okay, so this is where we stopped. Last time we were talking about the plasma membrane or the cell membrane, and I told you that I will be covering it up a little bit in detail when I talk about differences between bacteria and archaea. I would like to talk about the other layers of the cell envelope today. We'll move from outermost layer inward. So many bacteria, they secrete extracellular material outside the cell wall, which forms the outermost covering of the cell. This layer is called, the general term here is glycocalyx, and the glycocalyx, it can be differentiated based on the morphology. Let me just move this. Based on their morphology as well as based on the ease with which they can be separated from the cell, they are of two types. Either it could be a capsule or it could be slime layer. Now, the capsule, it's a distinct, very tightly bound layer formed majorly of water and polysaccharides, except in, for example, Bacillus anthracis, it is made up of polyglutamate. The capsular polysaccharides, they are covalently linked to the cell surface of the bacteria, hence cannot be easily removed. While in some bacteria, there is only one structure of capsule, many others form capsules which are quite diverse, structurally quite diverse, even within the same species. And the role of the capsule, well, it confers virulence or it adds pathogenic potential of the bacteria, it makes it all the more pathogenic. What actually happens is that the capsule protects the cell from immune recognition, the host immune response. On infection of a mammalian host, it is not recognized as foreign particle. It also protects the cell from the outer environment, for example, prevention of desiccation, other role in protection. It aids in attachment to surfaces. In fact, I don't know if somebody of you is aware of it. It's the capsule of the Streptococcus pneumoniae, that is the causal agent of pneumonia, that enhances the ability of the bacteria to cause disease. So non-capsulated strain of S. pneumonia, it either does not cause any mortality, or even if it infects, it's less pathogenic. There's a classical experiment based on this organism and its capsule called as Griffith's transformation experiment, which we will touch upon when we discuss bacterial genetics, or maybe before that you will cover it in your one of your biology classes. In contrast to the capsules, the other type of glycocalyx is called slime layer. Now, as the name suggests, it is slimy, unorganized, and is easily sloughed off from the cell as there are no covalent attachments with the inner layer. It is rich in moisture, so it protects the cell from desiccation. It also protects the cell from cell wall degrading enzymes and functions in adhesion to surfaces. It even serves as a reservoir for nutrients by trapping them. The next layer after the glycocalyx is the cell wall, which is the complex mesh-like structure that maintains cell shape. Now, because it is absent from eukaryotes and it's a necessity for bacteria, it is one of the most sought after target for selectively killing prokaryotes by using antibiotics. And we'll see the mechanism of some of the antibiotics in a while. In natural conditions, prokaryotes live in a fluctuating environment. The environment might be dilute, which would lead to accumulation of solutes inside the cytoplasm. That eventually leads to high solute concentration inside compared to the environment. So this generates a high osmotic pressure against the plasma membrane, which is delicate. It is a plastic structure, as you saw last time. So it must have an outer covering, which is the cell wall, that is porous, rigid, and has a high tensile strength. Now, together with being rigid enough to maintain high internal turgor and to protect the cell from external pressures, it also needs to be flexible enough to allow for expansion of cells when the cell grows. The material which makes this possible is a polymer called as peptidoglycan, that is a component of the bacterial cell walls. Peptidoglycan or murine is a polymer which is a backbone of two alternating sugars. These are both acetylated sugars, NAM, N-acetylmuramic acid, and NAG, N-acetylglucosamine. The sugar residues are linked by beta-1,4-glycosidic linkages. And the lactoyl group of each NAM residue, N-acetylmuramic acid residue, is substituted by a peptide branch, hence the name peptidoglycan. So there's a peptide component and a sugar moiety. These peptides have alternating D and L forms of amino acids. In most cases, the first amino acid is an L-alanine. Now the peptide branches, they are cross-linked either directly or by an interpeptide bridge. The cross-linking of the glycan strands generally occurs between the carboxyl group of the D-alanine at the 4th position and the amino group of diamino acid at position 3 of the peptide bridge. The peptide bridges are highly diverse in terms of composition and sequence and are sometimes even considered as signatures for particular bacteria. Now the polymer strands, the NAM-NAG residues together which forms this polymer, they are arranged helically, and with cross-linking it leads to a mesh-like arrangement. The chain length is also typical of a bacterium, varying from about 3 disaccharide units, disaccharide when I say it's NAM-NAG residue together, to about 250 such units. there are two features or maybe three features which makes it unique, this peptidoglycan unique. One is the presence of these unusual sugars, NAM and NAG. And then there is also the presence of the non-protein amino acids. Now, when I say non-protein amino acids, these are amino acids which do not form a part of proteins in a cell. I'm not sure if you know this, but the D form of amino acids are not otherwise found in cell proteins. In fact, this has a significance as a protective mechanism of the prokaryotic cell against proteolytic enzymes which a cell makes to break bonds between L forms of amino acids. Now, penicillin, for example, kills bacteria by inhibiting the transpeptidase which cross-links the peptidoglycan in the cell wall of a bacteria. Similarly, lysozyme also acts upon the cell wall. It breaks the glycosidic linkage, the beta-1,4 linkage that is between the NAM and NAG residue. Now, based on the content of PG or peptidoglycan, the bacterial world is divided into two groups. There are either gram-positive bacteria or gram-negative bacteria. You can see there is a difference in color. There are violet-colored gram-positives and pink-colored gram-negatives. Now, where does this word gram comes from? It comes from the Danish bacteriologist Hans Christian Gram, who way back in 1884 devised a test to detect if a bacteria had peptidoglycan. Basically, it was a test to see if the bacteria has peptidoglycan. In fact, his protocol is considered to be the most important staining technique in microbiology. It's used as a first test for identification of bacteria. Those bacteria that stain violet, like I said, are called gram positives, and those which stain pink are gram negative. Now, in this figure, you can clearly differentiate between gram positives and gram negatives. Can one of you from our last lecture, can you tell me what kind of shapes do you see here? Please raise your hand. Can you see? Can you tell me what are the shapes called?
Bacilli and cocci.
कौन सा कोकाई है कौन सा बेसलाई है कलर बोलो वायलेट एंड पिंक
Ma'am, the pink one is bacilli and the purple one is cocci.
Okay, good. Now let me take you through the protocol of gram staining, and this is important, students, because you'll be performing it almost on a very regular basis, okay, even for your other courses. Here what happens is, this will also be in fact one of the first experiment for general microbiology. Cells are first, the bacterial cells are first fixed onto slides and then treated with the primary stain, which is crystal violet. Then they are treated with iodine, which serves as a mordant. So it complexes with the primary stain. This is followed by decolorization using either ethanol or a mixture of ethanol and acetone. And finally, there is a treatment with counter stain, that is safranin. So the cells which retain the violet color, the second step that we had after fixation, it was the first step. They are termed as gram positives, while the one that take up the counter stain with safranin, which is pink in color, are called gram negatives. Now, what is the mechanism behind this differentiation? Gram staining is actually based on the ability of bacterial cell wall to retain the primary stain, that is the crystal violet dye post the solvent treatment. The cell walls of gram positive bacteria have a higher peptidoglycan and a lower lipid content compared to the gram negative bacteria, and so they can retain the first stain and are visualized as violet. Gram negative, as you can see in the figure, have a thin layer of PG, and in addition they have a lipid layer as the outermost covering of the cell wall and are unable to retain the primary stain. Hence, they are visualized as pink for the second stain that is added. Now, we will look deeper into the cell walls of both gram positives and gram negatives, and then we will come back to the grams protocol to relate it. Okay. The gram positive bacteria, they have a thick cell wall with several layers of PG as in peptidoglycan. There is an inconspicuous space between the plasma membrane and the PG layer called as the periplasm. Here you will find exoenzymes secreted by the gram-positive bacteria. Perpendicular to the PG sheets are polymers of teichoic acid, which are unique to the gram-positive cell wall. The teichoic acid polymers, which are anchored to the plasma membrane, for example here, are termed as lipoteichoic acid because they're attached to the lipid components. Now, what are teichoic acids? These are anionic linear polymer of either polyglycerol or polyribitol phosphates, which may be glucosylated or even amino acid esterified. Now, teichoic acids, they give the gram-positive cell wall an overall negative charge due to the presence of phosphodiester bonds between the monomers. They also help in adherence of the bacteria. This is as far as the gram-positive cell wall is concerned, which is the simpler one out of the two. The gram-negative cell wall is complex. It is composed of a very thin layer of PG, approximately 10 nanometer in contrast to 80 nanometer that you saw in the gram-positive cell wall. Sometimes here it's even just single layer of PG, but it is surrounded by an outer lipid layer. It has some similarity even with the plasma membrane, if you can notice. The peptidoglycan here is sparsely linked or branched as compared to that found in the gram-positives. Now, the PG layer is anchored to the outer lipid layer by lipoproteins called as Brown's lipoproteins. You will notice that in gram negatives compared to gram positives, there is a conspicuous periplasm. This periplasmic space contains hydrolytic enzymes, it contains various proteins, sugars, vitamins, other micronutrients, etc. The outer lipid is more permeable than the plasma membrane due to the presence of such structures called porins. Now, porins are channels which permit small molecules. In addition to the proteins and the phospholipids, the outer membrane of gram-negative bacteria contains a unique component, like you had teichoic acid there. Here, there's a unique component called as lipopolysaccharides or LPS. Now, the LPS are composed of three components. There is a distinct lipid A, a core polysaccharide, and an O antigen or a O polysaccharide. There are several roles of the LPS molecules. Lipid A acts as an anchor into the plasma membrane of the cell. It is made up of two phosphorylated glucosamines with the long chain fatty acids attached to both the sugar moieties. The chain length and the number of acyl groups of the fatty acids vary between bacterial species, but they are relatively conserved within a species. The core polysaccharide, the oligosaccharide of LPS, contains 1 to 4 molecules of another unusual sugar called as KDO, deoxy-D-manooctulozonic acid, and there are several hexoses in this core polysaccharide or oligosaccharide. It is joined to lipid A. Now, the third part is the O side chain or the O antigen, which is attached to the core and is comprised of repeating units of 3 to 5 sugars. This is the chain that you actually see. The O antigen has a high degree of structural variability as well, even in terms of the number of repeating units, this is quite variable. Now, the LPS serves many roles. It contributes to the negative charge on cell surface, just like you saw teichoic acid also conferring negative charge on gram positives. It may contribute to the attachment to surfaces, even biofilm formation. Then it leads to a selective permeability barrier because of the presence of these porins. Also, the lipid A of the LPS molecule is toxic and is considered as or even called as endotoxin that generate a strong immune response when the bacteria infects animals. Now, having told you about the structure, let us come back to the protocol of gram staining and what actually happens to the two types of cells. You will have to ask me questions if you don't understand, okay? Now, both type of bacteria are stained by the crystal violet, which is the primary stain. So you see them taking up the violet color. Addition of iodine, which is a mordant, leads to the formation of crystal violet iodine complex so that the dye is trapped inside the cell. So if you miss out on the step of iodine treatment, what happens is when you do the subsequent washes and treatment, the crystal violet which is entered the cell will exit the cell and you will no longer be able to see the violet color. So this process is called as fixing the dye. It's a mordant. Okay. Then upon adding the decolorizer, which is ethanol, or like I said, it could even be a mixture of ethanol and acetone, the outer lipid layer of gram-negative cell is dissolved. And you already know that the peptidoglycan is very thin and sparsely branched in the case of gram-negatives. So what it does, it leads to decoloration of the gram-negatives. In contrast, the same solvents dehydrate the thicker gram-positive cell wall, closing the pores as the cell wall shrinks during dehydration. As a result, what happens is the leaching of the violet iodine complex is prevented and the bacteria still stains as violet. Finally, a counter stain is added to the color, to the cells, and the gram negatives would take up this color and stain pink. क्या समझ में नहीं आया पूछो? Any step, anything related to why a particular step is there in the protocol? What happens if you miss out maybe a step? कोई मुझे बता सकता है? This is a widely accepted staining protocol from 1884. हाँ?
Excuse me.
बताओ.
Excuse me. Am I audible?
You are audible, but I don't know who you are because I cannot see the list right now. Tell me your name first.
It's Kashish.
हाँ कशिश बोलो.
Ma'am, why do we need to do the counter stain step? Ma'am, because in the decolorization step, the gram-positive bacteria is violet, whereas the gram-negative bacteria is decolorized. So why we need to do the counter stain step?
अगर तुम काउंटर स्टेप नहीं करते तो इस स्लाइड में जहां एक मिक्सचर है ग्राम पॉजिटिव और ग्राम नेगेटिव का, क्या तुम विजुलाइज कर पाते ग्राम नेगेटिव्स को?
Ma'am, they
कलरलेस
will
हो
be
जाता ना,
decolorized. So,
तो कलरलेस हो जाता ना.
अच्छा, okay, so they would not be
हाँ,
visible.
to visualize you will need to have some stain, some sort of stain. And ये देखो जो second stain है, जो counter stain है, it is lighter in color than the first stain. तो ऐसा नहीं है कि सैफ्रनिन को ग्राम पॉजिटिव्स लेंगे नहीं, लेकिन वो शो नहीं करेगा बिकॉज द डार्कर कलर विल डोमिनेट. ठीक है, समझ में आया क्यों काउंटर स्टेन चाहिए?
Yes, ma'am.
और कोई स्टेप, कोई मैकेनिज्म, ये इंपॉर्टेंट है, बहुत बेसिक है. विशंतन पूछो, लेकिन ये समझना बहुत जरूरी है कि कोई चीज क्यों हो रही है. Ask me, Vishantan.
Ma'am, I wanted to ask, do we only have like two sorts of bacteria which
Yes.
show gram positive, gram negative?
Yes,
No third
all
one.
bacteria, हाँ, all bacteria show either, देखो या तो पेप्टिडोग्लाइकन होगा या फिर स्पार्सली वो होगा, थिनर लेयर होगा, है ना. Other than mycoplasmas, which are actually without cell wall, every bacteria has a PG layer. अब वो डिपेंड करता है कि PG layer thick है, अगर thick है और आउटर सेल वॉल में लिपिड नहीं है तो वो ग्राम पॉजिटिव है. अगर वो ग्राम नेगेटिव है मतलब PG बहुत इनकंस्पिक्युअस है और आउटर एक लिपिड लेयर है. ठीक है, तो ऑल बैक्टीरिया एक्सेप्ट माइकोप्लाज्मास दे हैव अ सेल वॉल.
Okay, thank you, ma'am.
एनीबडी एल्स? बाकी सबको क्लियर है कि ये क्यों हो रहा है, क्या मैकेनिज्म है? ठीक है, अगर सबको क्लियर है तो बहुत अच्छी बात है. So after having covered the cell envelope, let us now talk about the appendages that prokaryotic cells possess. Now there are two types of surface appendages that are commonly found on some bacterial species. They are the flagella which are organs of locomotion, and pili. Now there's a discrepancy between in the nomenclature here. Some scientists use the terms pili and fimbriae interchangeably, while others consider the two to be two different structures. Now flagella occurs on both gram-positives and gram-negative bacteria. Mostly you will find them on bacilli and only rarely on cocci. In contrast, you will see that pili occur mostly on gram-negatives and are found on very few gram-positive bacteria. Now, there are various arrangements of the flagella, the way they are distributed on a cell, based on where and how many flagella are present. The suffix trichus, which means filament, is used to describe the arrangement. There are 4 types. There could be one flagella at one end called as monotrichus. There could be amphitrichus arrangement wherein there are flagella at either ends. Then there could be a tuft of flagella at either one or both ends called as lophotrichus. Lopho is a tuft. While there could be flagella distributed throughout the surface or perimeter, it's called as peritrichous arrangement. Bacterial flagella, they are long. They could be from 3 to 12 micron long and approximately 12 to 30 nanometer in diameter. It rotates like a propeller and it consists of 3 parts. The long filament that you see moving here, which protrudes from the cell surface. Then there is a hook which is anchored into the cell envelope. Now the flagellar filament is made up of proteins called flagellin. The protein subunits, they assemble in a helical manner to form a cylindrical structure with a hollow core. The movement of flagella is driven by the thrust generated from the counterclockwise rotation of the basal body, which is an energy requiring process. Now the gram-positive and the gram-negative cells, they differ in the structure of the basal body because of the difference in their cell walls. Gram-negative flagella usually have an L ring. L stands for lipopolysaccharide layer of the cell wall. A P ring, which is in the PG layer, peptidoglycan layer, and an MS ring that is located within the cytoplasmic membrane. There's also a C ring which extends into the cytoplasm. So the numbering is based, or the naming is based on the layer in which they are found. If you notice, there is a rod which is a major component of the flagellar basal body, other than these protein rings that I just talked of. On the other hand, in the gram-positive, you will notice just the 2 rings embedded in the plasma membrane. Looking at the other appendages, both pili and fimbriae, they are thin hair-like appendages on the surface of many bacteria. Pili is formed of the protein pilins. Pili are more rigid in appearance than the flagella. And what I've done is in the next 2 slides, I've presented a comparative of flagella and pili and fimbriae and pili for you to be able to differentiate them. Now we'll come to the last structure of the topic that is in the prokaryotic cell, which is the endospore. Now under adverse conditions, some bacteria, they form a dormant and tough structure called as endospore to evade the stress. So it's a survival strategy for bacteria. And endospores can survive stresses which could range from high temperature, high UV radiation, desiccation, chemical damage, etc. The extraordinary resistance that endospores exhibit makes them all the more interesting because they are not prone to being killed by many antimicrobial treatments. The endospore has a very special structure with multiple layers which confers this resilience to them. The outer coat, which is made of protein, offers chemical and enzymatic resistance. Now following the coat is a thick layer of PG called the cortex. The PG here is less branched compared to the regular PG that you would see. Now beneath the cortex is the spore wall with a regular content of PG. This eventually becomes the cell wall of the bacterium when the endospore germinates. Then there is the cytoplasmic membrane, followed by the center of the endospore called as the core. This is in a dehydrated state and has only the absolutely essential components, that is the cell's DNA and the ribosomes. Besides, it also has large amounts of what is called as dipicolinic acid, which contributes to the sturdy nature of endospores. There are small acid-soluble DNA binding proteins called as SASP, SASP for small acid-soluble DNA binding protein, which protects the DNA from damage under the harsh conditions. Now, how is the spore generated? This is a complex process requiring several hours. As a cell enters into the formation of an endospore, it divides asymmetrically by the formation of a septum, resulting into two compartments, the larger mother cell and the smaller pre-spore. This is at the stage 2 that you see here. Then the PG in the septum is degraded and the pre-spore is engulfed by the mother cell, forming a cell within a cell. This is the stage 3 in the figure if you see. Hence the term the endospore, forming inside a cell. So the spore is formed inside the mother cell. Subsequently, there is synthesis of endospore specific compounds, formation of the cortex, and deposition of the coats that you saw in the last slide. And finally, there is complete dehydration and maturation of the endospore. The mother cell then bursts and the endospore is released into the environment. Once favorable conditions return, the endospores germinate. That's about the sporulation cycle and how it then germinates. I think with that I have covered almost all the structures of a prokaryote. What I have deliberately missed out on is the cytoskeletal elements that are found in prokaryotes, which I will be taking up with the eukaryotic cytoskeletal elements, because I would like to tell you what is the analogy between the two. Other than that, now I'm open to questions. I don't think there are any other structures that I need to cover, any further structures. Now tell me, what are your questions from the entire unit, the prokaryotic cell, if there are any questions. ये भी बताओ कि कितना कवर किया फिर बैक्टीरिया में बायोलॉजी की क्लास में? आयुष वर्मा, ओके, ख्याति, सत्यकम,
Yes, ma'am.
tell me what all was covered in your bacteria, in the topic bacteria in biology, the structure or some other metabolic processes as well?
In the SBL, ma'am.
Correct, in the SBL course.
Ma'am, only structure is being covered till now.
The same structures that we are talking of?
Yes, ma'am.
अच्छा, तो मतलब repetition है तुम लोगों के लिए. What about archaea? Did she go into any further details of archaea?
Not as such repetition, but like here we are studying in a more detail. So.
तो वहाँ क्या cover कर रहे हो फिर?
Ma'am, there also we are covering the same topic, but not in much detail, right? I guess.
अच्छा. And so you will now move to the eukaryotic cell there as well, I guess. How is the course planned? Kashish? बताओ, क्योंकि मुझे भी यूकैरियोटिक सेल करना है ना.
We didn't
Kashish?
algae there.
एल्गी
Yes, ma'am.
करना है या कर रहे हो अभी? कशिश रानी बताओ.
Ma'am, abhi we'll cover it. In
which
the
algae?
coming classes. That I'm not sure of.
अच्छा,
I
because
think green algae शायद कर रहे थे.
तुम लोग शायद कैसे बोलते हो? मतलब कर रहे थे तो कर रहे थे. उसमें तुम लोग श्योर कैसे नहीं हो?
Because it was a end topic, so I think it will be covered in more detail later.
अच्छा, मतलब बैक्टीरियल सेल हो गया है, राइट? और यूकैरियोटिक सेल में तुम लोग ने रिप्रेजेंटेटिव ये लिया है, एल्गी लिया है.
Ma'am, you
Also did yeast.
Ma'am, actually, there was this. Ma'am started with this blue-green algae, and then there were some other topics, and then she started with the topic algae only. So that's why there's a little confusion.
सेल नहीं किया है सच, उसके सारे ऑर्गेनल्स वगैरह नहीं किया है.
ma'am, we were taught about protists, like it just began, and then we went to multicellularity, and उसमें मैम ने volvicine green algae का example दिया था. So like
अच्छा ठीक है,
it's
तो
just
मतलब इस तरह से नहीं. ठीक है, तो फिर मुझे यूकैरियोट्स में सारे स्ट्रक्चर्स आपको बताने होंगे.
yes ma'am.
एंड उसके बाद हम लोग रिप्रेजेंटेटिव फंजाई और रिप्रेजेंटेटिव एल्गी. सो बेसिकली आई वाज आल्सो प्लानिंग ऑफ टेकिंग अप अ ग्रीन एल्गा, बट उसमें उसके स्ट्रक्चर के साथ लाइफ साइकिल भी कवर करना है. दैट आई थिंक इज नॉट हैपनिंग इन योर एसबीएल कोर्स, इज इट?
Yes, ma'am. Ma'am, it's not being covered in SBL.
ठीक है सत्यकम. Okay, now any questions related to the prokaryotic cell structure, please ask me, because I'm not going to start the eukaryotic cell structure as of now. सब कुछ क्लियर है? ठीक है, क्लियर है तो फिर हम एंड करते हैं क्लास. चलो. Okay students, bye. You may